Now, without much further ado, I'd like to introduce our speaker for today. Our speaker for today is Professor Sally. Professor Sally is the Love Family Professor in the School of Chemical and Biomolecular Engineering and the School of Earth and Atmospheric Sciences at the Georgia Institute of Technology. She earned a doctorate in chemical engineering from the California Institute of Technology and was a postdoctoral scientist at Aerodyne Research Incorporated. Professor Sally's research focuses on understanding the chemical mechanisms of aerosol formation and composition, as well as their effects on health. Our group combines laboratory chamber studies and ambient field measurements to study aerosols using advanced mass spectrometry techniques. Professor Sally serves as the editor-in-chief of ACS Ascent later and is a member of the editorial board of ACS Earth and Space Chemistry. Professor Sally's research contribution has been recognized by the Sheldon K. Friedlander Award and the Kenneth T. Whitby Award from the American Association of Aerosol Research, the Ascent Award from the American Geophysical Union, the EPA Early Career Award, the Health Effects Institute Walter A. Rosenwich New Investigator Award, and the NSF Career Award. Professor Sally is currently leading collaborative efforts to establish the Atmospheric Science and Measurement Network in the United States. Thank you, Professor Sally, for joining us. The stage is all yours.
So today I'm going to talk about our secondary organic aerosols from biogenic and biomass burning emissions in terms of linking chemistry to climate and human health. And so this photo that you're looking here is a very hazy Atlanta. You see a lot of trees, so there's quite a lot of aerosols that can be formed from these natural emissions from trees. So we're going to learn about what happens when the trees are alive and what happens, when there are fires that burn the trees. So I'll just give a quick introduction on aerosols and particulate matter just to kind of get all of us on the same page. So aerosols are also referred to as particulate matter. The particulate matter with a diameter smaller than 2.5 micron is often referred to as fine particulate matter, or PM2.5. And these particles or aerosols or particulate matter have many different sources. As you can see here from the photos, you can come from vehicle exhaust, from wildfires, even from cooking, from ocean spray aerosols, also from trees. And so they are kind of really everywhere and indoors and outdoors also. And why are we interested in studying aerosols? So there are several major reasons, and one of them is health. So there have been studies estimating millions of deaths worldwide can actually be attributable to breathing in these PM2.5 pollution. And many times when you see those in the news, you see all these hazy pictures of cities and rural areas covered in smog. And the diagram here shows the global burden of disease. So you can see that out of the different reasons that cause mortality and morbidity, you can see that the air pollution is ranked very high in this global burden of disease and leading to a lot of human health and ecosystem health problems. And another reason we're interested in studying aerosols is their major impacts on climate. So this figure is from the IPCC report. It shows the different radioactive forcing agents. So you can see that these greenhouse gases, they are positive radioactive forcing because they absorb radiation and warms the earth. So what about aerosols? So aerosols actually play a very interesting role in climate change. So aerosols can impact climate by absorbing or reflecting radiation. They can also impact climate by seeding, the formation of clouds. And if you look at these charts, they are negative. So overall, the aerosols are cooling the earth. But the issue here is that if you see these uncertainty bars, there's a really large uncertainty associated with the impact of aerosols on climate. And so that's why we are interested in learning more about the aerosol sources and as well as their chemical and physical properties. So my focus today will be on secondary organic aerosols. But before we get there, let's have a quick introduction on organic aerosols as a whole. So you can see that there are different sources. You got trees, you got fires, and this could be from wildfires, could be from prescribed burns, could be from crop burning, just any sort of burning vegetation, and from cars, from vehicles, from industry. And so these different sources can emit aerosols as a whole. So if something is emitted as a particle, we call it primary organic aerosols. But at the same time, these different sources can emit gases. And these gases, we call them VOCs, which stands for volatile organic compound. So these gases, if they are emitted from trees, we call them biogenic. If they are emitted from human activities, we call them anthropogenic. So what happened to these gases in the atmosphere? So the atmosphere was very oxidizing. So once these gases are emitted from these different sources, they can be oxidized by different oxidants in the atmosphere. So these are, I guess, common oxidants in the atmosphere, including hydroxyl radical, ozone, and nitrate radical. And once these VOCs are oxidized, they can form different products. And so if the products are very volatile, then they will stay in the gas phase. But if the products are semi-volatile, then they do not want to be in the gas phase. They want to be a particle. So if you're a gas, but you want to be a particle, how do you become a particle? So if you don't have any particle next to you, then you can undergo nucleation and form your own particle. But the easiest way is actually to just condense onto existing particles. So these gases can undergo these process called gas particle partitioning and get themselves on the existing particles. And anything that is formed from this process is called secondary organic aerosol. And you can see from here, they are secondary, not because they are not important. They are secondary because they are formed from all these chemistry in the atmosphere. And so in my group, we use a systematic approach to study organic aerosols. So we conducted laboratory experiments in these chamber facilities to look at aerosol formation at a fundamental level. We also do a lot of ambient field measurements going around different parts of the world to characterize the aerosol composition and processing. And we make use of a large suite of advanced online mass spectrometry, including the aerosol mass spectrometer shown here, and this is a Figaro chemical ionization mass spectrometer. So in this talk, I'm going to tell you 3 different stories related to trees and also emissions from trees burning from biomass. And so the first story I'm going to tell you is between the trees and the vehicles. So trees emit a lot of different compounds. One of the major class of compounds emitted by trees is called monoterpene. So monoterpene is these C10H16 compounds. These are 3 major monoterpenes. They are very nice smelly. So if you go to a forest, and a pine forest, what you are smelling of the smell of the pine forest is actually alpha-pinene and then beta-pinene. The question is, what happened when these trees are planted in an urban area when there are a lot of cars? So cars can emit nitrogen oxides, including NOx, NO, and NO2. So when the emissions from the trees get oxidized in the atmosphere, right next to the emissions from cars, very interesting chemistry can happen. And so first of all, I'll show you the schematic of this aerosol mass spectrometer that we use pretty extensively in our research. So this is a schematic. You can see a puff of aerosol going into the instrument. You got a chopper, then you have a time of flight region, and then the aerosols are analyzed by the mass spectrometer. This is how it looks like as a photo. And so this instrument provides real-time continuous measurement of submicron non-refractory aerosols. So these are the 5 classes of compounds that it measures: organics, sulfate, nitrate, ammonium, and chloride. And I underline nitrate because for a very long time, we thought of the nitrate measure as inorganic nitrate, like ammonium nitrate. But there have been many studies showing that the nitrate can actually also be organic nitrate. So basically, when you use an AMS to measure nitrate, you can deconvolve the nitrate into inorganic and organic. And there's quite a bit of organic nitrates around, especially in the warmer months. And if you're interested, you can take a look at these publications. So we do quite a bit of measurements with the aerosol mass spectrometer in the Southeast US to understand how the emissions from trees and the emissions from cars can interact to form secondary organic aerosols. And to our surprise is that in addition to just the Southeast US, that we find a lot of organic nitrates. So you can see that organic nitrates are actually pretty prevalent in many parts of the world. So these pie charts shows the percentage of organic nitrates in the total organic aerosols. And you can see that in some places, the organic nitrates can contribute up to almost 80% of the organic aerosols. And there are not many measurements, as you can see here, from the Asian area. So this review paper was published in 2017. So if you have an aerosol mass spectrometer or other instrumentations, it'll be interesting to see what kind of organic nitrate contributions are in other parts of the world. So the question is, how are these organic nitrates formed, and what happens to them, after they are formed? So I'll quickly, walk through the organic nitrate cycle. So the photochemical cycle of NOx will make ozone. So ozone is a major pollutant, and it's also a major oxidant in the atmosphere. And ozone can react with NO2 to form nitrate radical. And nitrate radical is also an important oxidant, and is mostly at night. So as I mentioned, the atmosphere is very oxidizing. So once you have the VOCs emitted from different sources, these VOCs can be oxidized. These VOCs, which is the volatile organic compounds, can be oxidized at night, can be oxidized during daytime to form organic nitrate, which is this, RONO2. And as I mentioned about the secondary organic aerosol formation, some of these RONO2 can undergo partitioning. So now this is an aerosol, right? So it goes from the gas phase to an aerosol. And so it can contribute to, secondary organic aerosol formation. The question is, what happens, when these organic nitrates are formed? They go into the aerosol. So if the aerosol has water, it can undergo hydrolysis. Also, these organic nitrates can be photolysed, and they could release the NOx back to the atmosphere. And so when they release that NOx back to the atmosphere, it can undergo the cycle again and make ozone. So we are very interested in learning about what happens to these organic nitrates. So if you're interested in the hydrolysis, I'll refer you to these papers to read more about them. And for today's presentation, I'm going to focusing more on the photolysis of these organic nitrates and their impact on ozone formation. So to answer this question, we came back to the Georgia Tech Environmental Chamber facility, and this is a photo of the facility. Basically, it's an atmospheric simulation facility with controlled temperature and humidity, and also solar radiation. So there's all these UV lights that simulate the atmosphere. And so, as I mentioned, we are coming back to study this photolysis of monoterpene nitrates. So this is a schematic of the chamber facility. You can see that we have 2 reactors. So each of them is about 13 cubic meters. And surrounding the facility is a big suite of analytical instruments. So that would allow us to characterize the VOCs, the oxidation products, the aerosol formation, and the aerosol chemical composition, including this aerosol mass spectrometer that I mentioned earlier. And we also have a newer instrument called the FigoroSims. And FigoroSims allows for speciated measurement of gas and particle phase organics at the molecular level. So the AMS measures bulk composition, and the FigoroSims measure speciated composition. And so together we can learn a lot about the composition of the species that is generated in the experiments. So monoterpene organic nitrates, unfortunately, you cannot buy them from the shelf. So we have to make them. And so I have this former postdoc, Yuchen. So she did a lot of synthesis work to synthesize these organic nitrates from alpha-pinene, from beta-pinene, and from limonene. And these organic nitrates cover different functionalities, including primary, secondary, and tertiary organic nitrates. And you can see that they also have alcohol groups, carbonyl groups, and of course the nitrosyl groups, attached to them. And so we use these synthetic standards to have a better understanding of how fast these organic nitrates are photolysed. And once they are photolysed, do they actually impact ozone formation? So how do we do these experiments? So first, this is just an example of so this is 2 means secondary limonene hydroxynitrate. So this is just how limonene hydroxynitrate look like. So we inject them into the chamber. So now once this limonene hydroxynitrate is in the chamber, what happens to it? There are many things that can happen to this compound. So first of all, even if it doesn't do anything, it can just be lost to the chamber wall. Or if there's hydroxyl radical in the chamber, then it will react with hydroxyl radical. It can also react with ozone and be lost. And of course, our interest is to study, what happened with photolysis, right? So there are, there's different competing pathways that this compound can be lost. So how do we actually tease out, the photolysis rate of this compound? So I'll walk you through a typical experiment that we do in the chamber facility. So first of all, as I mentioned, we first inject the compound into the chamber, right? And you can see that it decreases a little bit. And so this can come from vapor wall loss, because as I mentioned, even if you don't do anything, given enough time, the compound will just be lost, to the wall. And then we turn on the light, and you can see that there's much more loss of this compound, because now this compound can be lost to the wall, or it can be consumed by ozone, or it can be photolysed. So the different processes are contributing, to the loss process that we observe here. And then we turn off the lights again. So now this part in this period D2, there's no light, so there's no photolysis, right? So now this compound can only be lost by just to the wall, or it can be lost by reacting with ozone. And then we just, for interesting observation, we also do this period D3, in which we add NO. So NO consumes ozone. So now if in D2, we have vapor wall loss and ozonolysis, once we add NO, then there's no more ozonolysis. So this comes back to, vapor wall loss. So you can see that these are different phases of the experiments. We got, 4 different steps. But if you look at these 2 steps, between the light period and also the dark D2 period, you can see that the differences of these 2 periods is the photolysis. So basically, based on the decay of the compound in these 2 periods, we can calculate the photolysis rate as the difference between KUV and also KD2. So this is how we back calculated the photolysis rate of different monoterpene organic nitrates. And we systematically conduct the same experiments for different organic nitrates. And it is very interesting to find that the photolysis lifetime of the organic nitrates is very short. So it is on the order of several hours. So basically, once these organic nitrates are being formed, they can be actually photolysed pretty quickly and release that NOx, back to the atmosphere. And so we attribute these short photolysis lifetime to the presence of nitroxy group and double bond or carbonyls next to the, which are very good chemophores. We can absorb the radiations and undergo photolysis. So coming back to my first question in terms of the NOx cycle. So with the photolysis of organic nitrate, you can potentially release that NOx. And this NOx can participate in the ozone formation again. So we did a quick model simulation to ask ourselves, will there be actually ozone being formed if this organic nitrate is photolysed? So this is just an example of an alpha-pinene hydroxynitrate. And so this is the time when we photolyze this compound. And we can see that quite a bit of NOx is actually released. So this is the gray area which NOx is released. A lot of it got converted to gas phase nitric acid, and some of them stays as a small amount of organic nitrate. And ozone, you can see that ozone steadily increases over time. And you might see that, oh, this is only like 0.34 ppb. That is not a lot. But bear in mind, this is PPT levels of this organic nitrate. And in the atmosphere, there are hundreds and thousands of different organic nitrates from monoterpenes and also from other VOCs. So this study really shows that it is important to understand, first of all, how much nitrate in your area is organic nitrate, and what are those organic nitrates. And when they get photolysed, they can impact ozone formation. So in terms of simulating ozone formation and understanding ozone formation, one has to consider organic nitrate. That is the main takeaway of this story. So now the question is, what happened when fires happen, when the vegetation is burned? So this is now my second story of today, which is the secondary organic aerosol and brown carbon formation from biomass burning. Biomass burning can cover the burning of forests. It can also cover, crop, burning, which could be a major problem in many other parts of the world. So this is a photo of Atlanta last summer. So Atlanta, we don't have a lot of wildfires, but if you remember from the news, last summer we were impacted also by the Canadian wildfires, who went all the way, from Canada, all the way from the northeast, from different parts of the country, and then come to Atlanta, which is in the southeast of US. So wildfires emit a lot of particles. And if you might remember from the first slide, if a particle is emitted as a particle, it's primary particle, right? But at the same time, wildfires also emit a lot of volatile organic compounds. So now this time, they are not the monoterpenes, because the monoterpenes are the nice smell, compounds from trees. But now the wildfires emit a lot of VOCs called furans. So furans are these, 5-member ring compounds. This is a phosphorol, this is a 2-methylfuran, this is a 3-methylfuran. And these can travel for a long time and get oxidized in the atmosphere. So we are very interested in learning what happens to these, VOC emissions from biomass burning. So again, we came back to our chamber and do a series of laboratory experiments. So again, this is your chamber here. And first of all, we inject some seed particles into the chamber, and most of the time we use ammonium sulfate. And the reason is because ammonium sulfate is a very common, type of inorganic aerosol formed everywhere around the world. It's kind of hard to find a place without ammonium sulfate aerosols. And then, of course, we inject the furan compounds. So we inject phosphorol, 2-methylfuran, 3-methylfuran, depending on which one we want to study. And we inject an oxidant. So this is a photooxidation. So we have a source to make hydroxyl radical. And then we turn on the lights. And then chemistry happens, and we make secondary organic aerosol. And of course, we have many instruments surrounding the chamber facility to look at the aerosol composition and optical and chemical properties. We also take filters, to look at them offline. So this is the temporal evolution of a kind of a typical experiment. So you can see that this is a 3-methylfuran here, so that the 3-methylfuran reacted away with an oxidant. And then at the same time, you're seeing the formation of organics and organic nitrate. So this is a pretty, I would say, typical experimental profile. And as I mentioned, we also collect the filters. So it is very interesting to see that the filters looks completely dark brown. And so what happened is that the photooxidation of furans form a lot of these light absorbing compounds. We call them brown carbon aerosols. And so we measure the mass absorption efficiency of these different SOAs. So this, you see the phosphorol, 2-methylfuran, 3-methylfuran, and the dry and humid is just whether, we do experiments under dry or humid condition. And so this MAE is I would say, highly widely used parameter to characterize the optical properties of light absorbing aerosols. And you can see that this figure shows the MAE of different types of aerosol, and you can see that the phosphorol aerosol, which is this one I'm highlighting here, has the highest MAE out of all the different compounds, followed by 2-methyl and 3-methylfuran. So as I mentioned, we have a large number of instruments that allow us to characterize the chemical composition of these aerosols. So what we find is that there's a strong correlation between the MAE and the nitrogen-containing organic compounds measured by the aerosol mass spectrometer, almost like a linear relationship. And so these nitrogen-containing organic compounds are either CHN or CHON compounds. And when we look at the composition of the aerosols more by looking at the offline chemical analysis, we find that there's a wide range of nitrogen-containing organic compounds. And I'll just highlight two major ones, which is the amine here and also the amide here. So these are many reduced nitrogen organic compounds. And so based on these measurements, we were able to propose a chemical mechanism in terms of how these brown carbon aerosols are being formed. So you can see the phosphorol here, and phosphorol reacts with the hydroxyl radical, and it can form this compound, and all these can undergo gas particle partitioning and reacting with the nitrogen in the ammonium sulfate seed, which is either ammonia or ammonium, to form these compounds. And because we have an online mass spectrometer, so we can actually see these compounds form in front of our eyes, that when we start the reaction, you can see that these red and blue compounds, they form pretty quickly, and then the aerosol formation is also observed. And these compounds can undergo further reaction in the particle phase to form a multitude of nitrogen-containing compounds. And so what happened is that these nitrogen-containing compounds, they have the CN bond, which can enhance light absorption. And so that's why we are seeing all these aerosols, completely looking brown on the filters because of the contribution from these nitrogen-containing organic compounds. And so this study really tells us that in terms of the biomass burning from wildfires, from prescribed burns, or even from crop burning, they are not just impacting the area around the burn. Many of these volatile organic compounds can get transported downwind, and they can form these brown carbon aerosols, which have major impacts on climate because they absorb the radiation. So now I'm going to move on to tell you, the last story. So as my title is Climate and Human Health, right? So now the next question we ask is that if these brown carbon aerosols or aerosols from biomass burning has these impacts on climate, could they also impact human health? So we undertook several projects to look at the toxicity of different types of organic aerosols. So this picture here shows a macrophage cell. So the macrophage are the first line of defense against any environmental insult. So we use this cellular assay to study the toxicity of different organic aerosols. So to answer how toxic or not, different organic aerosols are, we conducted an integrated chemical and cellular study in Atlanta in the summer and winter. So we deploy all our mass spectrometers at a measurement site in 2 seasons. And so for these instruments, we allow for characterization of the aerosol chemical composition. So remember the AMS give us the bulk, aerosol composition. The figurosym give us the speciated composition at the molecular level. And we also collect filters to analyze them with offline mass spectrometry and also FTIR. In terms of the health analysis, so we make use of this macrophage assay in which we collect the filters, we extract them, and then we expose to the macrophage cells. And this allows us to capture the reactive oxygen species generated from the cells. And this is a very comprehensive assay because it allows us to capture not just the oxidative, like the chemical activity of these reactive oxygen species, but also what is produced by the cells. And this is an important aspect to understand because oxidative stress has been widely proposed as a reason for the adverse health effects for breathing in these particles. So first of all, I'll share some information on the composition first before we go to the cell studies. So with the aerosol mass spectrometer, we were able to characterize the aerosol composition in summer and winter. And you can see these different species I mentioned earlier in the talk. And so one takeaway message is that everywhere is really organics. So sometimes in Atlanta, we have almost 90% of the aerosols are organics. So we are really, heading to a world full of organic aerosols. The question is, what are these organic aerosols? So we do a source apportionment analysis on these organic aerosols. And basically what we did is that we separate the organic aerosol into different types by using this source apportionment analysis. And so you can think of these bars here, they are coming from the green of this fraction. So let's take a look at the different colors of the bar. So now we are not just talking about organic aerosols, we are talking about different types of organic aerosols. So we can resolve biomass burning aerosol, cooking. As I mentioned, cooking is also a major source of aerosol, especially in cities. We've got this little gray area, which is the hydrocarbon light organic aerosol. They are from vehicles and also fuel combustion. We have the isoprene aerosol, so this is another emission from trees, and you can see that is only in summer because there are not many trees in winter here. But my focus today is actually on these 2 types of oxygenated organic aerosol. One is less oxidized, the other one is more oxidized. And so we call them LO and MOOA. And you can see that the LO is this lighter color here, the MO is the darker color here. But you can see that they are very important because they are almost 50% or more of the total organic aerosols. So what are these? If you remember my introduction slide, the organic aerosols can be primary or secondary. So it turned out that these OOAs, they actually surrogate for SOA. So what this figure shows here is that secondary organic aerosol really dominate the organic aerosol at least in Atlanta and also in many parts of the world. So we'll just remember these terminologies. We'll come back to it in the next slide. So as I mentioned, we use the source apportionment analysis to separate the organic aerosol into these different types. And so what we are interested in learning is to link these types to the toxicity measured by the cellular assay to learn about which type of aerosol is the most toxic to cells. And to do this, we conduct a multilinear regression analysis using the composition measured by the aerosol mass spectrometer and also the macrophage cellular assay. And we also include the transition metals in our analysis, but I'm not going to focusing on those today. So this is the multilinear regression analysis result. As I mentioned, there are a lot of things to go through, but I would just draw your attention here to the more oxidized and less oxidized OOA, as you've seen in these red boxes here. And you can see that no matter whether it's in summer or winter, you can see that these two organic aerosols are consistently determined as the major and positive predictors for cellular ROS. And this is especially true for this more oxidized organic aerosol. And you can see that there's a very strong correlation between this more oxidized OOA compared to cellular ROS. And so this is important and intriguing because if you remember the bar charts from before, the OOAs are really major, dominant fractions of the OA, right? So if they are very toxic, that is actually not good news. So we are trying to understand what compounds in OOA actually drive their toxicity. So to do that, we turn to our online and offline measurements. So with the FigurOSIMS, which is, remember this instrument that allow us to measure organic aerosols and gases at a speciated molecular level. We measure hundreds and hundreds of compounds. So what we did is that we separate the compounds into 4 different groups based on their degree of unsaturation. And then we also plot them against the carbon oxidation state. So the carbon oxidation state is just to show how oxidized the aerosol is. So you can see that this gray box here shows the range of the carbon oxidation state of the MOOA and LOA in the atmosphere. And now you can see that the LOA is less oxidized because the lower carbon oxidation state, the MOA is more oxidized because it has a higher carbon oxidation state. And what we see here is also with the compounds that are highly unsaturated, when we look at the composition, like this is a lot of information here, but I want to just, focusing on the aromatics. So what we see here is that for the compounds that are really having a high degree of unsaturation, many of them are also aromatics. So from this figure, you can see that if you follow my cursor here, you can see that there's really the strong correlation in which the highly unsaturated species are also highly oxidized. And remember, the highly oxidized species are OOAs. And we also did FTIR analysis, and we showed that there are a lot of acids and carbonyls in these compounds. So what happens when an acid and a carbonyl are attached to a conjugated or an aromatic system? So there's a lot of aromatics here. So these can be forming these compounds that the resulting, like all these different functionalities coming together can result in the formation of compounds that are really, redox active, and they can contribute, to cellular ROS. And so, one of the major takeaways from this is that the highly oxidized organic aerosol, when they have a lot of aromatic rings and also are unsaturated, they can really contribute to toxicity. And the bad news is that many of the biomass burning secondary organic aerosol have this type of functionality. So I'll quickly wrap up before I show the last couple of slides in terms of the 3 small stories that I told you today. So the first one is from the trees. Remember the monoterpenes from trees, which is the biogenic, and the NOx, which is from cars. So there can be interactions if you plant a tree, next to a car, that their chemistry cannot be, they're not separated. And so when monoterpenes get oxidized in the presence of NOx, you form a lot of organic nitrate aerosols. So it will be important for you to look at your maybe measurements and ask yourself, how much of your nitrate is actually organic nitrate. Because what we found is that these organic nitrates have very short photolysis lifetime, and so they can actually get photolysed and release the NOx and make ozone. So now we are not just talking about trees and NOx, we are also talking about ozone formation. So it is important to learn and understand to what extent the organic nitrate photolysis can impact ozone formation in your area. And the second story I mentioned is in terms of these photooxidation of furan compounds from biomass burning. So what we found is that the oxidation of these compounds can result in the formation of brown carbon and also secondary organic aerosol. And these brown carbon comes from the nitrogen-containing organic compounds, including amines and amines. And so the impact of a wildfire of a biomass burning event is not just in that area, but as the VOCs get transported downwind with these furans reacting away, you're forming aerosols that have major impacts on climate because they can absorb, the radiation. And at the same time, they can also have major impacts on health. So we learned that the oxygenated OA is the largest contributor to cellular ROS. And this is the major part of aerosol in pretty much everywhere around the world, is that most of the aerosol is actually very oxidized. So what we learned is that if the aerosol is highly oxidized with unsaturated species containing CO groups and aromatic rings, these compounds can really contribute to the cellular toxicity. And so if we think about this, as I mentioned earlier, what chemistry makes this type of compounds? So combustion, whether it's fossil fuel combustion or biomass combustion, make a lot of these compounds. So with climate change and with more burning happening, then we are going to make more SOA with these type of compounds. So it is important to recognize that SOA could become more toxic per mass in the future, resulting from larger contributions of biomass burning. So I urge everyone who are looking into this chemistry to kind of try to link your chemical measurements to some health studies to learn more about the toxicity of SOA. So I have a few slides to introduce a very new and exciting project called the Ascent, which is the Atmospheric Science and Chemistry Measurement Network in the United States. So this is a new opportunity to characterize aerosol sources, composition, and their impact on climate and human health. So this is the first long-term advanced ground-based measurement of aerosols in the US. So we have 12 sites, and this is a $12 million project supported by the NSF mid-scale research infrastructure here. And this is a site mapped, and you can see that we have different sites across the country. So we have a site here in Atlanta, and so the red sites are more of the urban area, and the green sites are remote or rural areas. So this really covers different geographical locations with different aerosol sources and impacts. And for each site, we have 4 instruments to capture the aerosol composition and properties. So the first one is the aerosol chemical speciation monitor. So this one is a simpler version of the aerosol mass spectrometer that I mentioned. We have an ethanometer to measure brown carbon and black carbon. We have an exact to measure metals, and we also have an SMPS to measure the size distribution. So together, this will really allow us to conduct real-time measurements of so many aerosol composition and property understanding. And so I just kind of want to flow back to this paper that I wrote more than a decade ago. So this was the work that I did when I was a postdoc at Aerodyne Research. So at that time, we are developing this ACSM for real-time monitoring, routine monitoring of ambient aerosols. And so this is really builds the foundation of the Ascent project. So it is really exciting that, after more than a decade, we have this opportunity to establish the Ascent network. And with more changes in emissions, in energy, in land use, it is now more important than ever to actually have the capability to measure aerosol composition in real time. So we'll be very excited if there are opportunities to collaborate with other networks in different parts of the world. So together we can learn more about the aerosol impacts on climate and human health in the long term. So lastly, of course, a lot of this work is done by the postdocs and students who are very talented and motivated. So I also highlight the 3 recent postdocs and graduate students who greatly contributes to the work that I presented today. And many of them are now, faculties and in their home country. So of course, I thank NOAA and NSF for funding, and thank you for your attention. So this is all I have today. I'll be happy to take questions.